Hello, everyone. It's about 17:30 here on the East Coast of the United States, and we'll get started. Just a reminder to everyone on our media advisory, this is an academic-only forum. Unpublished data will be presented here, and under no circumstances should this be shared on any social media. We are moving towards a first of the month, first Saturday of the month schedule. Our next conference will be on the 5th of September, and you can see the other dates there. Meetings of this webinar will be sent out within 72 hours. And to join the distribution list, please contact Susan Cordes, whose email is listed there. Next. I am Jeff Burns. I am the Chief of Critical Care at Boston Children's Hospital and Professor of Anesthesia at Harvard Medical School. I do not organize this by myself. I'm helped by a great deal of colleagues from around the world, and especially my two editorial assistants and colleagues, Megan Barrett and Susan Cordes. Next. Here's the agenda for tonight. We have a full agenda of some terrific speakers. The Q&A portion may be truncated because of this. We will in September move to a format where we will invite many of these speakers back to have a dedicated portion of Q&A, but the information that's being presented tonight is so important that we felt we should put this agenda together as it is here, and I will introduce each person as we get to the respective section. Next slide. It's a privilege to introduce my colleague, Dr. Lara Shekardamian, who is the section head of critical care at Texas Children's Hospital and a professor of pediatrics at Baylor College of Medicine. Lara last updated us 5 weeks ago. And Lara, can you tell us about the situation in Texas? Next slide. Lara, you're on mute still.
Thanks, Jeff. Hi, everybody. So we've been busy in Texas. I'll just give you a snapshot as I did last time, but obviously the numbers are very different. Nearly 500,000 people in Texas have now been diagnosed with coronavirus, and that's with a population of 29.9 million. Just to compare, the UK in the low 300,000s with more than double the population. And you can see here the daily reported cases really surged towards the end of August, early July, and it feels a little like we might be seeing a plateau, but a little bit early to start relaxing. And the bottom graph just shows the counts of positive patients according to age group, and the first 3 bars are the children aged 0 to 1, then to 5, and then to, so the last bar is 10 to 19, and that's out of 35,000 closed cases. So really the numbers, if we go to the next slide. Thank you. And this is the Houston data. The numbers are really around about 10% of all cases are pediatric. And this just gives you a breakdown by age group. And you can see just over nearly 5,500 children have been diagnosed in Houston out of nearly 50,000 now confirmed cases. So it's been very, very busy in the last few weeks. As I said, we and the adult units around town are seeing what's feeling like a little plateau with a little less frantic demand for beds. And I think one marker for us is the fact that we had taken around 10% of our admissions were adults actually with COVID at one point. And those requests for transfer have actually gone down. So it does feel like things are settling out. Could I have the next slide, please? This is the Texas Med Center hospitalizations by week. As you can see, we peaked right around the end of June and early July. And those of us who live in the US are breathing a little bit of a sigh of relief that we didn't see a surge in the sort of 10 to 14 days after July the 4th. And the next slide, please. And this is the TMC patients who are in ICU beds around the Texas Medical Center. That includes Texas Children's. And you can see huge numbers every day are admitted, but have been admitted. But again, we feel like we're in a plateau phase, but very, very different numbers to the early surge that the Med Center had in April. And then the next slide, please. A little bit about Texas Children's. We have added 3 COVID units to our original unit with a total of capacity of 37 beds. The most we've had have been 36 patients on a given day. We've had in the low 200s of patients admitted to TCH since COVID started hitting us. That includes about between 8 and 10% are adults. Around a third to 2/5 are ICU level, and that's a kind of vague number because our first and primary unit is an acuity adaptable unit, so we don't move out if the child gets sicker, we just scale up the level of care. We have had 3 patients on ECMO, including 1 adult. And I think just trends we've seen in the demographic, and this is to be borne out when we really look hard at our data, but certainly the uptick we've seen in June, July, we have seen older children, a lot of adolescents between the sort of 10 and 19 mark. One could speculate that might in part be related to Texas opening, and I guess the adolescents and teenagers going out a little bit more than the younger children. We are continuing to see MISC. We have had 20 patients to date, as at yesterday, in fact. We've had 1 of those has been on ECMO, and around a third requiring vasoactives, and around a quarter being intubated and mechanically ventilated. So that's our situation report. Thank you, Jeff.
Thank you, Lara, and we hope you and your colleagues are able to get through this as soon as possible. Next slide. I'd like to turn now to South Africa. I taped this interview earlier with Dr. Andrew Argent. Next slide. I'd like to turn now to our colleague, Dr. Andrew Argent. Dr. Argent is the chair of pediatrics at the Red Cross War Memorial Hospital in Cape Town. He's also a professor of pediatrics at the University of Cape Town in South Africa. Andrew, for many years, led the critical care program at the Red Cross War Memorial Hospital. Andrew, welcome.
Thanks, Jeff. It's really great to see you.
Andrew, we're all aware that the Southern Hemispheres are really bearing the brunt of the pandemic right now, and we've all been, of course, concerned about what's going on in Africa. Could you tell us, how has COVID-19 impacted children in Cape Town and the Western Cape area?
All right. Jeff, if I could share my screen, I can show you just some of that data, and we can just run through some of it. And obviously there would be time for comment and question after that. COVID in children and a Cape Town perspective. And this is a screenshot from the data that's available today from the Western Cape government, the Western Cape being the most southerly part of South Africa. And as of today, we've got nearly 94,000 patients. We've gone just over 3,000 deaths. And you can see in this slide on the right that our pandemic really kicked off at the beginning of May. We hit a peak somewhere around the end of June or early July. And now the numbers seem to be settling down a little bit. And so that's where we are overall as a pandemic. But I want to really focus a little bit on the pediatrics. So here we are at the moment. Of those 95,000 cases, about 5% have been laboratory confirmed in patients under the age of 20 years. So on the 24th of July, 88,000 cases on nearly 400,000 tests. That translated into nearly 23,000 hospital admissions and about 2,800 deaths. But exactly as you and others have presented in the US and in China, children have had relatively low numbers of infection. If you look by age group, under 20 years, we picked up some, sorry, I'm just getting the numbers wrong there. We had some 2,000 between the age of 15 and 19, right down to 855 under the age of 5 years. Now, obviously that's skewed by testing procedures and protocols. But I really want you to focus a little on this data. So if you look at the purple line here, that demonstrates the percentage of the population in different age groups by 5-year quarter groupings all the way through the age spectrum. And you'll see that for the Western Cape, a large chunk of the population, probably half the population, is under the age of 30 years, with a relatively small group of people who are actually beyond the age of 65 or 70. If you look at the number of patients who've been diagnosed in this orange line here, you can see that the peak of cases diagnosed is in this group of the 25 to 35-year-olds. And probably that relates to people who are active in the community, moving around, and possibly feel themselves less vulnerable. When you look to those who hospitalized, that has shifted, and you can see that the peak is now somewhere between 55 to 60 years of age. And when you look at the deaths in the black line, the peak by percentage of population is very heavily in the older group of the population. And so, again, I don't think that's terribly different from what's been described across the rest of the world, except the focus on the different distribution of population. How does that translate into numbers for children? So again, of those 90,000 patients by age group, we're looking at some 400 pediatric hospitalizations, and at the moment, some 14 deaths. When we start talking about the pediatric multi-inflammatory syndromes, we're talking of less than 25 to 30 patients diagnosed in the entire Western Cape population at this stage of our epidemic. And so the numbers are relatively trivial. Of course, for those individual patients, they needed careful care, and some of those have required intensive care and renal support therapy. But let's take that whole disease profile and talk about some data of life for children in our community. And so in the Cape metropolitan area, population probably of somewhere around 4 million people, we have 123,000 children who live in households where there's only one adult. It doesn't take very much imagination to realize that if that adult were to get infected, those children would be at extreme risk, because the older people, the grandparents who would normally step in to help, are scared to step in at this time. If you look at where those children are, some 32% of children with lone caregivers live in informal dwellings. And frankly, I'm talking about shacks where transmission rates are extremely high. And probably 40% of them live in cramped conditions with more than 2 people to a bedroom. In those conditions, social distancing just is not going to happen. And to make it even worse, in this small area, we're talking about before COVID, nearly 20,000 children were living below the food poverty line. And when you look at massive unemployment related to the COVID pandemic, you're starting to get a sense of just how children could be impacted. I think it's really important as we look across the world at this pandemic to remember the different population pyramids. This is South Africa, and you can see that the majority of our population are actually in that mid-range, somewhere between teenagers and 30, which correlates with the peak in our infections. By contrast, North America has a very different distribution of population. Europe shows an even greater preponderance of elder people in that population. Asia, little between both the Europe, US, and Africa. But that's what Africa looks like. That's sub-Saharan Africa with 1 billion people, and probably half of those below the age of 18. And that immediately starts to start talking about the profiles we're likely to see. And so I want to leave you just with a couple of thoughts, that for children, the actual infection with COVID may not be a cause of significant morbidity and mortality. That doesn't mean we don't have to look after children. It doesn't mean that they're not children who are at particular high risk. And it doesn't mean that there's some children who develop unique syndromes in response to COVID. But we do have to understand that it's going to be the social, economic, and environmental implications of this pandemic that have the potential to be utterly catastrophic for children. And as a community, as a global community, we really need to focus on the factors that will make a difference to child survival. It's quite possible that the epidemic will carry a different course in Africa. That's because of the population profile, the prevalence of poverty, and many other factors that I haven't spoken about in detail as to now. One other component that I could perhaps add in, that in a country like Ghana, I'm told that less than 10% of the population get a monthly salary. You can start to imagine the implications for income, for household security, food security, and for children when you implement a shutdown and people cannot go out day to day to earn their living. So I'm going to leave you with that, and Jeff, I'd be really happy to answer any comments or questions.
Well, Andrew, thank you for that absolutely insightful overview of the challenges that you're facing, which have nothing to do with PPE and enough ventilators, and has everything to do with basic health and food and secure maintenance of housing conditions. I think that your presentation will be especially relevant to our next speaker, who proposes that we should be relying more on far less expensive testing, such as antigen testing, in order to kind of control the pandemic. And the challenges that you're facing speak directly to that. Andrew, as you know, we're under a time limit here, but I hope that we can bring you back in the webinars coming forward so that we can understand how this has evolved for you. But thank you for everything that you're doing in Cape Town.
That's an absolute pleasure, and thanks for the opportunity to share with your group, and thank you for the work you're doing.
Yeah, next slide, Megan. I'd like to turn now, as I said, I taped that earlier with Andrew Argent, and we don't have time for Q&A as I discussed, but he's obviously illuminated for us the problems of sub-Saharan Africa, and for literally millions of people around the world that we don't confront in most of the countries who are listening here tonight. But I can only think of perhaps one other speaker to follow Dr. Argent for the work that he's doing, and for the novel approach that he's suggesting about how we should be doing surveillance for COVID-19 in order to suppress this virus in all environments. So it's a privilege to introduce to you Dr. Michael Minna. As you see here, Dr. Minna's got a number of affiliations: the Harvard School of Public Health, the MIT Broad Institute, the Brigham and Women's Hospital, and Harvard Medical School. Michael, welcome.
Thanks very much. It's a pleasure to be here, and I guess we'll just get started. So slide. So I'm going to be speaking today about different testing strategies that we've been evaluating. I'm going to start out by giving us a very, very broad overview of the differences in testing modalities. But then I want to dive in briefly into some newer analyses that we've been doing that I think can help to curb outbreaks where they're hitting populations the hardest. Next slide. So first of all, why do we need surveillance testing? SARS-CoV-2 or COVID-19, I don't think I have to tell anyone on this call, is not going away anytime soon. It only seems to be picking up speed in much of the world. And so over here on the left, we see the cumulative cases that have occurred. This is as of maybe a week ago or so. And daily new cases are still continuing to increase. And we know that SARS-CoV-2 is likely to be play by similar rules as its closest neighbors, which are the other seasonal coronaviruses. They have very strong seasonality associated with them. And what we can see on the right side is if you average over multiple years, you end up seeing that September, October, November, really, and December end up seeing large increases usually of seasonal coronaviruses. So in the Northern Hemisphere, that is. And so we really have to be careful, and we need to make sure that we have our surveillance system set up in a way that can not only just monitor what maybe is happening a couple weeks ago, but we really need to have a very good understanding of what's happening right now and who are most infectious, so that we can use surveillance as an actual tool to stop outbreaks. Next slide. So there's a few different approaches, but the two major sort of tests that we might think about doing for surveillance are virus testing and serological antibody testing. I'm not going to talk too much about this serological or antibody testing, but I do want to point out that virus testing is for the here and now usually. It tells you who is infected at this moment in time and can give you an opportunity to act and respond appropriately through contact tracing or any of the other roles. Serology, on the other hand, is a very powerful tool that I think has gotten, unfortunately, has been a little bit neglected throughout this epidemic because we've been in crisis mode more or less throughout the whole of the epidemic. But during peacetime, for broad surveillance of the population, antibody testing can be a very, very powerful tool and can actually and can serve to highlight and detect outbreaks very quickly when they are occurring from a public health perspective, and then potentially allows you to move in with virus testing when you detect an outbreak. So this slide is really just to suggest that serology shouldn't be neglected as a very powerful tool for public health, in particular during peacetime, once in locations where the virus has largely subsided and long-term monitoring is needed. Next slide. And this is just one example to depict that. On the left is virus testing, and on the right is antibody testing. What we can see is virus testing usually stays positive for a very limited amount of time when you actually have the virus in you. And so therefore, it's not always as powerful a tool given a given amount of testing that might be happening versus antibody testing. Once somebody becomes positive, what I'm showing here is on each row is an individual, and each column you could think of as a week in time. And what happens is after somebody does turn positive, they remain positive for at least months usually. And of course, there's been a lot of discussion about exactly how long the time is during a primary infection for SARS-CoV-2, but on average, it's going to be much longer than the virus remains positive. And so it can be a very powerful tool overall to identify outbreaks, which then you can move in and allocate resources appropriately to then stop those outbreaks. And so if there's other questions about this, I'm happy to talk about them later. But there's some more pressing, I think, new avenues for surveillance that I want to get to right now. Next slide. I'm sorry, I have two slides here that I think illustrate really how virus testing and serology can be very useful and powerful. And this is just to point out, if you have a virus test, like a PCR test, and you're using it at a population level, it tells you the here and now. It tells you who's getting infected currently, but it doesn't really give you much information about the epidemic trajectory that you're facing at the moment in time. So this is an example. If let's say you're in a town and you find that 5% of people are virus positive at that moment of time, you don't really know too much about what to make of that 5% positivity, except that you have an outbreak happening. But is it going up? Is it going down? It's a little bit difficult to know. Next slide, please. If you add serology into the mix, however, just a cross-section of positivity with a virus plus a cross-section of antibodies will give you a very powerful idea of the trajectory that the virus might be taking. For example, if you add serology and you find that 55% of the population is antibody positive and 5% are virus positive, you can have a pretty good understanding that the epidemic trajectory is on its way down. On the other hand, if you find that antibody positivity is only 5 or 6% and virus positivity is 5%, then that might be an area where you really want to allocate resources quickly to help mitigate what could become a pretty big outbreak. And so this is just, I do believe that serological evaluation can provide a massive amount of extra information, and it's very cheap, and so it can be scaled up quite well. Next slide. So I want to talk about surveillance characteristics and what are some of the trade-offs that we might need to make to really have surveillance that not only just gives some information to put on a website, but actually can serve in real time to stop transmission and curb outbreaks in a matter of weeks. Next slide. Surveillance test characteristics are usually, these are three of the main results that we might want in a test. We, of course, a lot of people think that we want a very highly sensitive test. We want to know who's virus positive when they're positive. And so usually this is perceived as a very high sensitivity of a PCR test to detect viral RNA. We want test results that are fast, and we want tests that are low cost, which can be done with very high frequency because the cost is low. The priority order that we normally place these in is usually sensitivity number 1, I'd say. And then cost and apparently speed to get results has been way lagging in terms of our priorities in this country, with many results getting to people weeks after they've been tested, which frankly, I think makes these tests essentially useless if you're finding out your results 7 days or more afterwards. Next slide. And so is this the correct order? A fast test that can return results in minutes, that is sensitive, but is expensive, would be great. But if it's too expensive, you're not going to be able to scale it up very well. You're not going to be able to get everyone tested in the country. But what about a test that gives you very fast results, is very inexpensive, but is 100 times less sensitive, or maybe even 1,000 times less sensitive than the PCR test? Is this okay? Is this a good trade-off? Most people would think no, but I'm hoping to suggest that there is actually a very reasonable argument to be made that cheap, fast tests that are less sensitive potentially are very powerful. Next slide. And of course, this is important right now because I believe that these tests can be available today. I don't just believe it, I know it. But there are important barriers in the way at the moment. Next slide. So to understand why I'm saying that test sensitivity can be reduced if you have a trade-off of frequency, I want to show this slide, and we have to look at the biology of this virus and how it grows inside of our bodies. Once somebody, on the x-axis, I have days since infection, and on the y-axis, I have viral load that might be in the body. And what I want to point out is once this virus, once somebody becomes, there's an incubation period, once somebody becomes positive on the PCR test, it's only a matter of hours usually, as the virus is in exponential growth. It's increasing very, very quickly, and it will surpass thresholds of low sensitivity tests very quickly, usually within hours, maybe within a day. And this is important because it's not really until the virus gets to very high copy numbers that it actually starts transmitting to people. So do we need to necessarily worry about having such a high sensitivity test that we can detect 10 viral RNA copies, or is detecting 100, 1,000, or 10,000 RNA copies as the limit of detection an okay trade-off if it means that you could have a daily test every single day? And I think that what that would ultimately do is it would allow you to detect when people are transmitting in time to stop them from going out of their house that morning, for example, stop the transmission. And what's very important here is that when we evaluate these tests and we say they're lower sensitivity, it's very important to know what our targets are and what it means. When we test these with a given sample of specimens on a given day, and we say that cheap tests or antigen tests are less sensitive, it's very important to understand that almost all of the samples that are being missed as positive are in this yellow period here. It's oftentimes weeks after the infection has stopped transmitting, and the RNA just lingers in the nasopharynx for a very long period of time. Meanwhile, the antigens are gone, the proteins have unfolded, and you just have residual RNA sticking around in double-walled vesicles inside of our cells for a long period of time, detectable on the PCR, but meaningless from an action perspective, and frankly, meaningless from a risk of severe disease perspective, because you've already gone past the most crucial period of time in terms of severe infections. And so we did some modeling based on this kinetics to understand what would it mean if we introduced a daily or an every 2-day or 3-day low sensitivity at the molecular level test. And what I'm trying to get at is, can we change our view from molecular sensitivity to our sensitivity to detect infectious people? And I think we just need to change the target for what we define as a appropriately sensitive test. Next slide, please. So we modeled this out using both agent-based models and more SIR ecological models that are a little bit more simple to deploy. But in both cases, we found nearly identical results. And ultimately, what I'm showing on the left here is what fraction of the infectiousness is removed given different types of tests. And in the pink bars are the current PCR type of tests. And in the darker bars are what I would call low sensitivity transmission detection tests. And what we can see is in both cases, the most important thing by far is the frequency of testing. Outside of frequency, there's almost no difference on the left between the high sensitivity test and a test that's 100 times less molecularly sensitive than the PCR test. And it really comes down to how frequently can we test people. And if we're doing testing daily or every 3 days, we more or less abrogate all or stop all infectious individuals from being out in the population at any given time. We drive R naught way down, or the effective R. And on the right side, we see the total number of infections compared to no testing. And frankly, I would say that at the moment, we pretty much have a situation where we have the equivalent of no testing in the United States anyway. And daily or every 3 tests with either sensitivity test, if not even weekly of most people, would drive down infections. And this suggests that frequency of testing is much more important than the sensitivity, than the ability of the test to detect molecules. Next slide. And not only is it the frequency of testing, but it's also the turnaround time for tests. Now, at the time when we wrote this, we didn't even consider making a turnaround time of a week or 2 weeks, which is really what we have now in many cases in the States. But we have, for each of these different testing frequencies, we have 0 means rapid turnaround time in minutes. 1 or 2 is 1 day delay, and 2 day delays. And we can see that relative to the rapid tests, even just having a 24-hour delay and definitely having a 48-hour delay, for example, if your frequency is every 3 days, greatly reduces your ability to decrease infections at the population level. So the best test, regardless of sensitivity, would be one that can be frequent, can give rapid results. And because this is a virus that inside the body grows across many orders of magnitude, potentially you get infected and you have a viral load of 10, you end up transmitting when you have a viral load of 1 million or 1 billion or 1 trillion sometimes we can detect in people. So this really suggests that when we're trying to understand sensitivity, our targets are the wrong thing. I don't think we should care if we can find 10 RNA molecules in somebody. We should care if we can find 1 million and stop it right there at the source.
So Michael, could I just jump in to make sure I'm following you? The gold standard PCR limit of detection is roughly 10 to the 3rd.
Yeah, depending on what units, but yeah, you could say 10 to the 3rd, yep. And
the people in the chat are asking, what does that correlate to on a CT value?
About 40.
And your argument is that the limit of detection for a point of care test or a self-administered test, which is cheaper, faster, less sensitive, is at maybe 10 to the 5th?
Yeah, 10 to the 5th, maybe 10 to the 6th even per mil, or a CT value maybe around 30 to 34.
And then the threshold for infectivity?
Probably much lower, probably a lot lower, especially if we consider who are the likely super spreaders, which are really the most important people to find. It's probably a threshold. So we know that at a CT value above 30, 32, 34, we can't even detect, we can't even culture the virus out of somebody with that. So it suggests that it's all just RNA with no virions. But there's even been some papers, I think in Nature Medicine, that showed that above a 24 CT value, there was no culturable virus. So it really suggests that you probably have to have, my guess is somewhere in the teens or below and up to maybe the mid-20s of CT values would be the most likely time when somebody would be transmitting. And this isn't actually, in my opinion, it's not a difficult question to answer. We actually have it if we just take 2 different data sets. We know what period of time people are transmissible, which is usually day -2 to day 4 or 5 after symptom onset. By the time you're day 10 after symptom onset, even the CDC says you can go back out into the world. So it's usually this small window of time. And if you look at the CT values of people from day -2 to day 5, it's usually very high, somewhere in the teens to low 20s. So I would suggest that it's much higher viral loads when people are most likely to be transmitting.
And so a reminder to the audience that the viral load increases as the CT value decreases.
That's right.
And one last question for you, and then not to slow you down. The time from going from 10 to the 3rd to 10 to the 5th to 10 to the 6th is a measure of a day approximately, or a measure of several days?
That's correct. If we can go back a few slides to that graph of the kinetics, I'll just point that, yeah, this one. So what I've highlighted in red here is that duration of time. And it's probably on the order of a day when you pass through multiple orders of magnitude. By the time you become PCR positive, the virus is in what we might think of as deterministic exponential growth upwards. And so it grows very fast, just like in a petri dish, and it can grow over multiple orders of magnitude in just a day. And so I would suggest that it's a very short, there's very, very few people that actually would get tested in that very short amount of time. And most people who are getting tested with our current surveillance system are out in the yellow time period there, in this very long drawn out period of RNA positivity.
Thank you. So please catch up. I just wanted to make sure I was following you.
Sure. Next slide, please. So I just want to conclude, and I'm going to have a few more discussion points, but I think that both serology and virus testing are extremely important and work well together, like I showed in the first couple of slides. But in order to rapidly detect and prevent outbreaks in a period of time that is useful to actually tell somebody that they need to stay home, our results say it's more than okay to sacrifice molecular sensitivity for speed and low cost or high frequency testing. And I would suggest that this means that we need to change, it's a pretty simple solution. We just change our target when we're evaluating what it means to be sensitive. Just detecting somebody who's PCR positive, I think, in many ways you could think of as usually a false positive. It's potentially weeks after somebody's cleared the virus for the most part. They're no longer transmitting and they just continue to have a positive. I'll show a little bit of evidence for that. But I do really think that it's time that we change our priorities from cost to make cost and frequency of test number one, and speed to get results should be within minutes and not days, and definitely not within weeks. We may as well, any lab that's giving back results in a week, I think we should just stop those tests and start to reset everything. There's no point to give tests back a week later. And number three, I think, should be the molecular sensitivity, with the first two essentially driving the notion that we want sensitivity to detect infectious people, not sensitivity just to detect 10 or 1,000 RNA copies.
And these would be saliva-based tests that you could do, be self-administered, be at home, not technically complex, saliva into some reagent. What's the cost?
Yeah, so if you go to the next slide here, I think I show that. Oh, sorry, I don't show it. But you can, I think that these things can be about $1 or so. I think it's in 2 slides and then I'm pretty much done. But on the next slide, I just want to point out, and we've now discussed it, but why don't we have all of these rapid tests today? They can exist for about $1 or $2 at a time. I think the federal government could be making these, printing millions of them if they took control of the situation. But currently, there was a lot of fanfare just a couple of days ago because the FDA posted a new template for at-home over-the-counter tests. I think it does very little to get us where we need to go. It does open the door to say they're open to it, but ultimately it still requires 90% molecular sensitivity against a representative sample set. And what I'm showing here on the bottom right, this is the last few weeks of virus CT values in our hospital here in Massachusetts. And what you can see is the vast, vast majority of CT values from week to week have been well into the upper 30s, meaning the viral loads have been exceptionally low. So if we were to take what the FDA considers as a representative sample set and try to measure the sensitivity of any of these tests right now, we would miss all of these. The sensitivity would look terrible. But that's because currently our CT values, our viral loads from people during surveillance is really very low. So we're just looking at the wrong target. Next slide. And so this is really what I think we need to go to. Right now, even with the new FDA reports in the RADx program and things like that, I think what will get approved are going to be like the Nespresso model of virus testing. These are going to be very sensitive, sleek, very good tests, but they're going to still be expensive. They're going to be highly manufactured and not really approachable to get to everyone in the country. They're going to require, the FDA is requiring that every negative result as well as positive has some way to get reported to the public health agencies. So we're going to end up bottlenecking ourselves into what they call rapid diagnostic at-home tests. But what I want is I don't want Nespresso. I just want something that's cheap enough that can get caffeine into everyone every day. And this would be a daily at-home paper strip you're seeing a picture of what they might look like. They might sit into like a little plastic container or something. You spit into it, swirl it around, and it gives you a positive or negative. but it's cheap. It's not connected to any Department of Health website. It's just paper. And that, I think, if we can get these out to the whole community, to millions and millions of people to use daily, which is reasonable to assume we could, it could actually stop outbreaks that are happening in Texas and Florida and Arizona overnight. I mean, practically within a week, I think we could drive the R naught way down below 1. I think this is my last slide. Next.
Michael, I'm sorry to say that we don't have a whole lot more time. As I told you, many people wrote me beforehand, and as you know, there are representatives from the WHO, the NIH, and the US and European CDC listening now. Your argument certainly makes a lot of sense to me. I've also seen you mention that we have excellent contract tracing in Massachusetts, but because we're relying on the PCR test, your argument is that they're not nearly as efficient as a public health measure as they could be with this kind of approach. Is that correct?
Yeah, in fact, I mean, just looking at those CT values that we get from Massachusetts alone, it suggests to us that the vast majority of positive results that we are detecting are probably weeks after infectivity has stopped. So we're putting all of these contact tracing resources into tracking down people and saying, look, the last 48 hours after their sample was collected, but in reality, they were probably infectious 1 or 2 weeks before their sample was collected. So we're just wasting contact tracing effort for the most part. And that's why I would also suggest that if short of these types of tests, we should really be using the CT values of the tests as a very strong indicator of who should get contact traced and what's the level of infectivity somebody might be at.
Well, the implications for opening school and the implications for really for what Andrew Argent was referring to of a better ability to suppress the virus until there's a vaccine. You make a very provocative argument, and we look forward to seeing the publication of the data that you presented, which I understand is, I know I saw on the preprint server, and I suspect is under review somewhere. So we look forward to seeing the publication of these data. And Michael, I thank you very much. I harassed you during your vacation, and thank you for joining us tonight. And I hope we can have you back in the future when we have a little more time.
Thanks very much. Pleasure to be here.
All right. And Megan, if you could accelerate us up to, there we go. There you go. To our next segment. It's been almost 15 weeks since we've heard from our experts in pediatric infectious disease. And we're delighted to have them back tonight to update us on the literature. As you see there, we have 5 international representatives: Dr. Katie Chattis from Children's Hospital of Philadelphia, Dr. Donna from the University of Padua, Julie Fitzgerald from Children's Hospital in Philadelphia, Dr. Liu from Beijing Children's Hospital, and Dr. Mary Nakamura from Boston Children's Hospital. Drs. Chiotis, Fitzgerald, and Makanura were the authors of the Pediatric Society of Infectious Disease guidelines on antiviral therapies, and they're here to update us on the literature. So Katie, I believe I'm turning this over to you. Next slide.
Hi, this is Mari. I'm going to actually start us off, and then Katie will take over next. Thank you very much, Jeff, for this opportunity to speak again. Next slide, please. We would like to start with some disclaimers. So we will be discussing current evidence-based therapies for management of pediatric COVID-19 patients, but we will not be getting into MISC in this presentation. We've included therapies for which there is evidence for or evidence against the proposed therapy, but we've excluded those for which the evidence base is severely limited. The following presentation is based on our expert consensus opinion. Much of the available evidence is preliminary, and all of it is from adult subjects, so generalizability to other patient populations and settings should be assessed. Next slide, please. To start us off, we wanted to provide some context by briefly summarizing recommendations from guidelines, representative guidelines from our respective countries. First of all, the Chinese guidelines are recommending in favor of glucocorticoids in severe cases, as well as immunoglobulins in severe cases, and traditional Chinese medicine. The guidelines are neutral on interferon alpha nebulization or interferon alpha 2b spray, as well as on the antivirals Arbidol and lopinavir ritonavir. They are against use of antibacterials unless there is an indication. Next slide. The Italian guidelines are in favor of remdesivir for patients who have severe illness, which is defined in the box below, as well as dexamethasone for patients who have ARDS or progressive deterioration of respiratory function, or for multisystem inflammatory syndrome. They're neutral on remdesivir for mild or moderate COVID-19, as well as IL-1 blockade, IL-6 blockade, and convalescent plasma. And they recommend against use of hydroxychloroquine or chloroquine, with or without azithromycin, lopinavir ritonavir, or steroids in patients who are not requiring oxygen unless there's another indication for their use. Next slide. The US NIH treatment guidelines are fairly similar to the Italian ones, but with some interesting differences. So they are similarly in favor of remdesivir for patients on supplemental oxygen and dexamethasone for patients who either require oxygen or mechanical ventilation. They note that hospitalized adults should receive VTE prophylaxis per standard of care. The NIH guidelines are neutral on remdesivir for mild or moderate COVID-19, but are actually also neutral for patients on the more severe end of disease, so those who require high flow oxygen, non-invasive or invasive ventilation, or ECMO, because of insufficient data. They are similarly neutral on convalescent plasma, IL-1 blockade, and IL-6 blockade, and then thrombolytics or full dose anticoagulation for VTE prophylaxis. The NIH guidelines recommend against hydroxychloroquine with or without azithromycin or chloroquine, lopinavir ritonavir or other HIV protease inhibitors, dexamethasone for patients who don't require supplemental oxygen, and JAK inhibitors. Next slide.
I'll take it from here. This is Katie. So to evaluate the various evidence-based COVID-19 therapies for children, we came to the following framework, which considers the published data, other published guidelines, as Mari highlighted, and collective expert opinion. The strongest recommendation is to recommend or recommend against, which essentially establishes the therapy as a standard of care. The next tier in our framework is to suggest, again, in favor or against, which means that the group assess that there's a sufficient evidence base weighting towards or away from a benefit of the therapy. Finally, in the middle, which unfortunately is where many of our recommendations lie, is a recommendation to consider a therapy, which reflects the uncertainty as to whether or not there's benefit of the therapy. Within this category, we've additionally indicated therapies that should be considered, meaning that based on what we know now, most would accept that the benefits outweigh the potential harms. And therapies that could be considered are those that really different choices may be appropriate in different scenarios and based on individual values and preferences on both the provider and patient side. Next slide. Here we've summarized the therapies we reviewed today, ranked using this framework. In the following minutes of our presentation, we'll step through an evidence summary for each and provide a summary of our recommendation for each of these therapies, which this slide summarizes. I'll also mention that the Journal of the Pediatric Infectious Diseases Society will soon be publishing an update to our original guidance document related to antivirals and an additional document that summarizes immunomodulatory therapy as well as convalescent plasma that highlights the literature supporting these various recommendations. Next slide. I'll start by discussing antivirals and starting with remdesivir. The first study I'd like to highlight is the Adaptive COVID-19 Treatment Trial, or ACT, which is an NIH-funded study that was recently published in the New England Journal and forms the basis of many of the recommendations surrounding the use of remdesivir. This randomized trial evaluated the impact of 10 days of remdesivir therapy on a primary outcome of time to recovery on an ordinal scale, which is reproduced on the right of the slide, essentially hospital discharge or ongoing hospitalization without medical need. Next slide. The key finding from this trial was that there was a reduction in time to clinical recovery in those patients who were treated with remdesivir, 11 days versus 15 days in placebo-treated patients. The other key takeaway is shown in the three figures, which summarize data from the overall cohort on the far left, with the subgroup just requiring supplemental oxygen at the center, and those recovering mechanical ventilation or ECMO on the right. So as you can see, the overall positive findings in the trial in favor of remdesivir seem driven largely by the subgroup on supplemental oxygen, where benefit was evident, with really no difference in time to clinical recovery in the cohort on mechanical ventilation or ECMO. Though we do need to be cautious in our interpretation of these subgroup analyses, reminding ourselves that definitive conclusions shouldn't be drawn based on these type of analyses. Before presenting our recommendations for remdesivir, I'll also very briefly highlight an additional key study, and that is the work of Goldman and colleagues who compared 5 versus 10 days of remdesivir in an open label randomized trial of hospitalized patients with severe COVID-19 disease, those on supplemental oxygen, and demonstrated no difference in clinical status as measured on a similar ordinal scale at day 14 following randomization with either duration of therapy. Next slide. So for children, I've summarized our key recommendations here. So for patients with severe disease, we recommend that remdesivir should be considered given the adult data supporting benefit on time to recovery in this illness severity. The second bullet point for those critically ill children, that is patients on invasive or non-invasive mechanical ventilation, our consensus was also that clinicians should consider remdesivir. We do acknowledge that this population did not demonstrate significant benefit in ACT, and also that the NIH guidance issues a neutral recommendation for critically ill populations, but we did feel that should consider was the most appropriate recommendation given the extreme illness severity, the overall ACT findings, and uncertainty of how the differences between adult and pediatric illness severity and natural history of disease allow us to apply the adult data to our pediatric cohorts. Finally, our group favored administration of up to 5 days of remdesivir therapy for most children, with the option to extend to 10 days in mechanically ventilated patients. And I want to emphasize the up to, as if the child is better and approaching discharge or significantly clinically improved, or you determine that there's another reason for their symptoms, people shouldn't feel compelled to treat the quote unquote full 5 to 10 days, as the benefits of remdesivir are really very, very uncertain in kids, and the harms, cost, and availability are real issues. Next slide. I want to move through the next 2 slides pretty quickly, as I believe that the scientific and medical communities have come to consensus that these next 2 therapies are unfortunately not effective in treating COVID-19 based on the data we have available to us. In the case of hydroxychloroquine, there have now been multiple randomized trials in hospitalized patients, as well as for post-exposure prophylaxis now, evaluating the efficacy of hydroxychloroquine with and without azithromycin, with no benefit in mortality, clinical outcomes, or virologic clearance demonstrated, including the recently published recovery trial or recently released recovery trial, which is a very large randomized trial performed in the UK. Several observational studies, which are of variable methodologic quality, admittedly, also support these findings. Therefore, we suggest against use of hydroxychloroquine or chloroquine with or without azithromycin outside of a trial. Next slide. Finally, I'll highlight the 2 largest randomized trials that have evaluated the use of lopinavir ritonavir for the treatment of COVID-19, and both have demonstrated no significant difference in mortality or virologic outcomes, leading our group to suggest against its use. Next slide.
I will take us through a review of the evidence for steroids as well as convalescent plasma. We suggest that corticosteroids should be considered for children who require mechanical ventilation, and that they could be considered for those who require supplemental oxygen. Based on the lack of evidence of efficacy, we suggest against use for children with no need for supplemental oxygen. Next slide. The key evidence on steroids come from the recovery trial, which Katie just mentioned. In the trial, about 2,100 adults received IV dexamethasone for up to 10 days and plus usual care, and were compared with 4,300 control subjects who received usual care only. The study findings were that for all patients overall, that the risk of 28-day mortality was significantly lower among patients who received dexamethasone. The risk of mortality was also lower for those who required oxygen or invasive mechanical ventilation, but there was no difference observed for those who did not need oxygen. Next slide. Our consensus on convalescent plasma is that it could be considered for children with severe or critical COVID-19. Next slide. Our best data on convalescent plasma come from a single trial. It was a randomized controlled open label trial conducted at 7 centers in Wuhan between February 14th and April 1st. In it, 52 adults who received convalescent plasma plus standard treatment were compared with 51 controls who received standard treatment only. Notably, due to lack of eligible patients after March 27th, the study was closed early with only 103 of the intended 200 subjects enrolled. Next slide. Overall, it was found that there was no difference in the primary outcome of time to clinical improvement. A subgroup analysis was performed by disease severity that suggested, as shown in the middle panel, possible benefit in severe disease, but a test for interaction by disease severity was not statistically significant, and so the investigators cautioned about drawing firm conclusions about that result. Subjects who received convalescent plasma did have significantly higher rates of conversion to negative SARS-CoV-2 PCRs at 24, 48, and 72 hours of evaluation. Next slide. I'm not going to cover this slide in detail, but we've included more information for those who would like to refer back later. The best evidence we have about the safety of convalescent plasma comes from an evaluation of convalescent plasma transfusion in 5,000 adults who were enrolled in the US FDA's Expanded Access Program. The evaluation showed that overall, the rate of adverse events was very low, so just 36 were reported within 4 hours of transfusion for an incidence of less than 1%. Next slide.
I'll be covering immunomodulatory therapy and anticoagulation recommendations. As with the other therapeutics that we've discussed so far, there are currently no immunomodulatory therapies with proven efficacy for treatment of COVID-19 in children. Our recommendation is that immunomodulatory therapies, and more specifically IL-6 blockade and IL-1 blockade, could be considered in children with confirmed critical COVID-19 who also have some evidence of hyperinflammation. In this situation, consultation with subspecialists who are familiar with prescribing these therapies and who understand the side effect profiles of these therapies is recommended. I also want to extend a word of caution, as all of the peer-reviewed data that exists on these therapies in the setting of COVID-19 come from adult observational studies, and thus they are subject to limitations in the potential bias of this type of study. I'll provide a brief update on the new evidence for immunomodulatory therapy in adults that has been published since the last time we spoke at this forum. Next slide, please. As I've previously presented, we know that there can be mild to moderate elevations of IL-6 in both adult and pediatric patients with COVID-19. These elevations have been shown to correlate with disease severity and mortality, but may be orders of magnitude lower than those seen in other hyperinflammation syndromes such as HLH or the cytokine release syndrome that is related to CAR T-cell therapy. Additionally, IL-6 is known to increase the risk of secondary infections. At the bottom of this slide, I included data from 2 separate adult retrospective cohort studies evaluating tocilizumab, an IL-6 receptor antagonist in COVID-19. The first study was performed in mechanically ventilated adults, and the second in hospitalized adults with COVID-19 pneumonia. Both studies showed a statistically significant lower hazard of death, but both showed higher rates of secondary infection with tocilizumab therapy. Next slide. Preliminary results of an industry-sponsored phase 2/3 clinical trial in the US for sarilumab, which is also an IL-6 receptor antagonist, were released by the sponsors. And this is preliminary data, which is encouraging in the patients with the critical category of COVID-19, but not in the severe COVID-19 group. The 3 treatment groups, placebo, low and high dose sarilumab, are shown in the column headers. The second red box highlights that there was lower mortality in the critical patients receiving the higher dose. And the bottom box, the bottom red box that I've highlighted shows that there's a higher discharge rate in the group that received the higher dose of sarilumab. Based on these results and the opposite trends that were seen in the severe patients, the company has announced plans to continue with the phase 3 trial only in the critical subgroup. We would still advise caution, however, as these are preliminary data from adult patients and the trial is not complete and has not been peer reviewed yet. Next slide, please. IL-1 blockade is also being explored, and I present here data from 2 adult observational cohort studies, both of which used historical controls receiving usual care. Anakinra is an IL-1 receptor antagonist, and the short half-life and the safety profile are favorable aspects of this therapy. The data are not as strong or abundant for IL-1 blockade as it is for IL-6 blockade, but the familiarity with this drug and the safety profile lead us to provide the same conclusion that it could be considered. Both studies here, adult studies, showed a lower hazard of death, and there was no difference in side effects in those treated with anakinra in these studies. Next slide. I included this slide as a reference for the audience to provide some early data on some other immunomodulatory therapies that are under investigation in COVID-19, but the data for these therapies are really too limited to provide recommendations for these therapies outside of the setting of a clinical trial. Next slide. Finally, we wanted to spend a moment on anticoagulation. The hypercoagulable state has been well described in COVID-19 with characteristic lab abnormalities and multiple putative mechanisms. Existing data support a lower mortality in adults with elevated D-dimer who receive prophylaxis for VTE, but no data exists currently to support using full dose anticoagulation as prophylaxis for VTE. Included in this, the highlights of the NIH guidelines regarding anticoagulation. I think that the main points to take home from this table are that, according to the NIH, the diagnosis of COVID-19 should not change our usual standard of care for VTE prophylaxis in hospitalized children. And to draw your attention to the last bullet point, that when caring for patients who are rapidly deteriorating, particularly in pulmonary, cardiac, or neurologic status, the possibility of thromboembolic disease that could be treated as a cause for this deterioration should be investigated rapidly. Next slide. Thank you for the opportunity for us to update our findings and to speak with the group today.
Thank you all for that wonderful overview. It's pretty evident that we're having a time problem here. And so here's what we'll do. I do definitely want to get in the announcement from the NIH. They do have a special announcement that they requested to make. And at the same time, there's a very large audience that undoubtedly wants to hear from the senior authors of the recent publications on the multisystem inflammatory syndrome in JAMA and the New England Journal. And I'm just afraid we can't do justice to both. And so Mike Levine and Dr. Adrienne Randolph are here. I don't know if Dr. Zucker was able to join us, but Mike, may I suggest that I invite you and Dr. Randolph back in September to do this segment. And at that time, perhaps we'll have some more information as well. I know I'm putting this on the spot in front of many hundreds of people, but how does that sound to you?
Absolutely fine, Jeff. It's been a wonderful evening to hear the talk so far, and very happy to present it at a later stage.
And let me offer this, Mike. You do have a slide on the BEST trial, and so Megan, could you advance forward to near the end of his presentation? And let me just give a minute for Dr. Levine and a minute for Dr. Randolph to each present on information on how to reach them about their various registries. And we'll start first with Dr. Levine's registry with the BEST trial. Now you can see why I thought it wasn't prudent to go back, if you could, Megan. Yeah, there you are. So Mike, you want to take a minute to remind people what the BEST trial is and how to contact you or the trialist to enroll a patient?
Thanks, Jeff. I think the fundamental aim of the Best Available Treatment Study, or BATS, is the concept that we desperately need randomized trials, but it's going to take a long time before we have any result from randomized trials in the pediatric inflammatory diseases. And we have many patients presenting, particularly in countries where the pandemic is only just taking hold, particularly Asia, Africa, and South America, and of course the United States, and we're expecting upsurges in many other parts. And there are now excellent statistical methods for correcting for imbalance in severity of patients. And if we can enroll all the patients that have already been treated throughout Europe, the United States, Canada, in this Best Available Treatment Study, we would be able to do an analysis of which of the treatments works. And what I was going to show in the previous slides is that we have absolutely no definite data as to whether immunoglobulin, steroids, or biologicals does improve the outcome. We have the clinical impression that they do, but we only have clinicians' best guess that the treatments that have been used up till now are beneficial. And so really this slide was my last slide to appeal to all clinicians who have treated patients with the pediatric inflammatory diseases, that if they can enroll in the study, it's a study that does not collect individual personal data. It only collects the institution and the age of the child. But if we can get all the patients who've already been treated by whatever treatment you have given, and in some cases, no specific immunomodulatory treatment has been used, we will be able to use propensity matching to work out which treatment actually improves the outcome as well as resolves inflammation most rapidly. And this is particularly of importance for countries where immunoglobulin and biologicals are not available. I was struck by Andrew Argent's talk right at the beginning of the very different situation in Africa and other places. If we knew that steroids were just as good as immunoglobulin or biologicals, this would be a huge help to those countries which don't have the resources to be using immunoglobulin and biologicals. So I think this is really an appeal to colleagues around the world who are seeing these patients to enroll them in the study. It's an online webinar. Everyone who enrolls a patient will become a member of the study group. We intend to publish the findings simply as from the best available treatment study group, and list everyone who's contributed patients and every center who's contributed patients. So this is a genuine international collaboration to try and work out what treatment should be used for this disease. Thanks, Jeff.
Thank you, Mike, and thank you for accepting the live offer to come back on September 5th and present. Dr. Adrienne Randolph, could I give you a minute to briefly describe what's going on with your CDC study in the United States?
Yes, so the Overcoming COVID-19 study is a study across the United States. We now are across over 70 centers. We have two parts, a public health surveillance registry where we collect very detailed data. And we also have a study with consent where we also collect patients' blood and respiratory samples. We have many aims in this study, but it is limited to the United States. We're doing a very deep dive on how patients are managed, treated, symptoms. There's a very detailed case report form. And we have collaborations with the Pediatric Heart Network, who is going to do follow-up of patients at about at least 20 centers and get better assessment of cardiac outcomes, led by Jane Newberger and funded by the NIH. And we're also partnering with other networks such as the CARA Rheumatology Network and others that we're working out agreements with that will allow us to, and the BloodNet, to look at coagulopathy in more detail so that we can do further investigation on these complications in these patients.
Thank you. And Dr. Randolph, could I also invite you back as a discussant with Dr. Zucker when Mike Levine presents on the MIS in September, and we will put information on your trial in the minutes as well. I want to turn now to our colleague from the NIH, Bill Kapayanis. Bill, as you see here, is a senior medical officer with NICHD, and he has a special announcement to make. Next slide. Bill, are you there?
Yes. Can you see me?
Yes. Yes, we can. We can see you and hear you.
Thank you for this opportunity to talk to you tonight about the notice of intent to publish several funding opportunity announcements for the RADx-RAD initiative, including the Prevail Kids RFA, which is quite relevant to this audience. Next slide. So just a little bit about the initiative itself. The rapid acceleration of diagnostics was made possible by supplemental appropriations that was signed into law by Congress in the end of April that provided $1.5 billion that was transferred to the NIH Office of the Director to develop and deploy diagnostic methodologies to rapidly respond to the COVID pandemic. Next slide. And so the goal is to accelerate innovation in development and commercialization of and implementation of COVID testing through funding of early innovative diagnostic technologies, the advancement of late-stage diagnostic technologies to expand testing infrastructure, the identification of effective testing implementation strategies in underserved populations who are disproportionately affected by the pandemic, and doing all of this by working closely and coordinating with key government agencies to secure approvals and also dissemination and scale up in the communities. Next slide, please. And so the RADx projects are numerous, and the investments are substantial. I won't go into any length on many of them here that you see, but only to say that some of them, like the RADx Tech at the top left, are beginning to bear fruits with some of the at-home or point-of-care testing that are hopefully going to be getting rolled out for the community to use in the ensuing months. So the difference with the RADx Radical program, which is what we're talking on tonight, which is this $200 million longer-term vision of what to do in the longer term to develop and advance further novel, non-traditional approaches or new applications of existing approaches to address gaps remaining in COVID-19 testing via the 7 initiatives that you've read in the notice of intent to publish. And that are also supported by a data coordinating center. These initiatives all involve unconventional screening approaches, biological and physiological markers, new platforms and point-of-care devices, as well as incorporating data science and AI technologies. You could read more about them here in this site, or you could turn to the New England Journal review written by Dr. Collins et al. About 10 days ago. Next slide, please. And so to go to focus on the one of those 7 initiatives, which we spearheaded with our colleague institutes, other 6 institutes at the NIH, and in partnership with the Office of the Director in developing the Predicting Viral-Associated Inflammatory Disease Severity in Children with Laboratory Diagnostics and Artificial Intelligence, or PREVAIL Kids RFA. Next slide, please. Next slide. And so this is because we know that the pediatric SARS-CoV-2 clinical spectrum varies from asymptomatic to mild in most cases, fortunately, but then in some cases progresses to moderate and severe and even pretty significantly severe, the multi-inflammatory, multi-systemic inflammatory syndrome, or MISC. And so next slide. This spurred us to encourage the development of novel approaches for case finding to understand this spectrum and to predict the longitudinal risk of disease severity after a child is exposed or infected by SARS-CoV-2, and to figure out how to best treat that child and optimize health outcomes. So the objective of the RFA is to support multifaceted, non-traditional innovation research, including novel testing strategies and technologies, for example, diagnostic and prognostic biomarkers and biosignatures used alone or in combination, as with artificial intelligence-based algorithms, to rapidly diagnose and characterize MISC associated with SARS-CoV-2 and to accurately risk stratify and predict disease severity throughout this spectrum. So, next slide, please. The scope that we hope to see studies address would hopefully include as many of these areas below as possible. So genetics, omics, and other biomarkers, viral dynamics, and immunoprofiling studies, digital health platforms could be incorporated, leveraged for children, of course, and artificial intelligence and machine learning to develop and test and validate predictive algorithms for disease severity. So the mechanism would be a phased award innovation mechanism that's 2 years per phase, and it could be up to 4 years per award. Of course, this is a milestone-driven transition process that advances only those testing strategies and technologies which have significant promise to lead to an accurate and reliable MISC prognostic algorithm for helping children. And so the data coordination aspects, we're going to be leveraging the infrastructure of the RADx RAD DCC. The funding, I was hoping to be able to share more with you, but next slide, please. As you will see, the RFA was slated to be released on Friday, and we have a slight delay, but it is imminent, and hopefully we'll be on the lookout very soon and to see that. And the due date for the applications are expected in September, with first award in December. There is a related FOA I'd like to mention with our colleagues from BARDA, that is the Pediatric Diagnostic Tools for Severe COVID Disease and MISC. The link there below you can access for more information and context if you need. And the next slide will go to my last slide, which is thanking you. And if you have any questions, please reach out to me, and I will end there.
Bill, thank you for that announcement. Everyone on this webinar, of course, is happy and thrilled to see that there's more investment in these disorders in children. Next slide. Thank you all. As I said, we're moving to the first Saturday of each month. The next webinar will be the 5th of September. You already know some of the agenda. And thank you for Dr. Levine and Dr. Randolph for agreeing to transfer their presentation to the 5th of September. And you see the other dates there. Minutes will go out in 72 hours. To join the distribution list, get the minutes, get access to the video and audio of this webinar, please contact my assistant, Susan. And any ideas about topics or speakers, please email me. And thank you all for joining us. And we look forward to seeing you on the 5th of September.